Hi, I'm Davy Weissenbacher, a research data scientist at the Cedars-Sinai Medical Center. I will present here our collaborative work with our colleagues from Arizona State University and the University of Pennsylvania. In this study, we aim to identify automatically PubMed articles in which the author reports the generation of new SARS-CoV sequences and using natural language processing. Our motivation for this work is that since the COVID-19 pandemic started, millions of SARS-CoV-2 genome sequences were sampled from patients worldwide and deposited in public databases such as GSAID and GenBank, but the sequences that were deposited typically did not include associated patient-related metadata such as the demographic of the patient, the disease severity, the comorbidity, or the outcome of the disease. And this lack of information limit their utility for genomic epidemiology, which is the field that studies the evolution and the spread of viruses in a specific population that are later used for guiding public health effort. The patient-related metadata associated with the sequences may be available in the article where the author reports sequencing the viruses, but with nearly 400,000 articles related to SARS-CoV indexed in PubMed, a manual correction analysis is not easily done. So here, as a first step, we present and evaluate a pipeline to identify this PubMed article, and we leave as a future work the extraction of the patient metadata contained in the article that we will detect. Our workflow is described in the main figure of the poster. We downloaded the LitCovid collection, which is a subset of PubMed articles related to SARS-CoV-2 that were automatically identified by the National Library of Medicine in the US using machine learning. At the time of the study, the collection contained 117,000 full articles, and we kept the articles that have a reference section to select only the full article and reject the articles that were limited only to their abstract in the collection. Then we segmented the sentence of the article using spaCy, and since the article mentioning new sequences were sparse in the collection, we sampled randomly 100 articles to define keywords to match those articles. We applied the keywords on the 117,000 articles and selected randomly 245 articles for full annotation. We re-encoded the sentence in the XML format to import them into the Inception annotation tools, where two annotators annotated each sentence of the 245 articles as evidence that the author reported generating new SARS-CoV-2 sequences versus something else. Once annotated, we split the article annotated into a training, a validation, and a test set, and we used the training and the validation set to train a BERT classifier to detect these sentences. Once trained, we applied the BERT classifier to the collection of the full article, and the classifier found 3,190 articles. And after a manual review of the prediction, we found that 1,621 articles were true positive, with at least one sentence where the author of the article reported generating new sequences. Although keywords help us to find articles of interest, making a complete list of keywords is unlikely and causing us to miss a large number of the relevant article. So training a BERT classifier was a way to automatically expand these keywords and enable us to find more articles. In detail, our keyword filters are described in Table 1. We apply the keyword sequentially, and the list contains words like extracted or synthesized, or the name of sequencing platform like PacBio. 75 articles in our 245 were annotated by both annotators to compute the entire annotator agreement, which was very high, with 1 point Cohen-Kappa score at the article level. That is, they both agree that at least one sentence in the article the author reported generating new sequences, and 71 at the sentence level. Our corpus is very imbalanced, with 74 articles reporting new sequences and 171 which do not, and this translates as the sentence levels as only 357 sentences labeled positive for 50,000 negatives. We trained a classifier on 196 articles and evaluated on 49 articles. We tried to undersample by training only on negative sentences that were similar to the positive sentences, but this did not help. We achieved our best score by training on all sentences available. On the test set, we achieved a limited score of 48 F1 score at the sentence level, but a good score of 80 F1 score at the document level, with our classifier being able to retrieve all articles of interest in the test set, while limiting the prediction errors to an acceptable rate of 67 precision. Evaluating on the test set only was not sufficient, since this set is biased. All the articles were selected by keywords. So we performed an additional evaluation. We deployed the classifier on the full collection and evaluated the precision by manually reviewing the true and the false positive, and we evaluated the recall by looking at a sample of 47 articles that were of interest because they are linked in GenBank to a genome sequence deposited in the database, with 12 found by our keyword and 35 were not found by the keyword. On this set, the performance slightly dropped to 64 F1 score with a precision of 50, and a recall of 85. 40 of the 47 were retrieved by the classifier. To conclude, using BERT will retrieve 30% more articles than using a standard keyword search. Our error analysis in the table below showed that to improve the performance, a classifier should consider a full paragraph and not a sentence. Unfortunately, our preliminary tests with large language model were not successful. Feel free to contact us if you have any question. Thanks.